I'm Alan Trounson from Cartherix, back at ARM after a long time, and good to be here and seeing many faces that I've known from when I was in California and when I was working at CERN. So this is a panel on customised solutions for genetics medicines, navigating the GMP supplier landscape. Pretty important area, and there's a lot of things going on that the panel, I think, can really inform, and it's a great panel. Panel members who are mic'd up can come up. For most of the translation research units, bringing a genetic medicine to the clinic requires support of several outside partners. Researchers need to procure custom GMP raw materials like plasmid DNA, guide DNA, Cas9 nucleases, as well as outsourcing GMP services like cell therapy manufacturing. All these services carry a cost and a significant timeline. From the end user perspective, it's necessary to balance these costs and timelines in order to enter the clinic and begin treating patients on time. From the supplier perspective, it's important to innovate and provide clients with flexible products and services. The purpose of this panel is to bring these two perspectives together. So both the supplier and the client perspective. So this is what we want to do. I'm going to really allow questions to come from the audience, so we would like you to participate with us. I don't want to sort of have a one-way discussion if it's possible. So all you need to do is get your paws up, and there'll be a microphone around. There's a big box over there that you can grab hold of, and so that everybody can hear the question, and particularly me hear the question. So we've got the panellists. I think we're all here now. So I'll introduce the panellists. Again, I'm Alan Trounson. I'm the CEO of Cartherix. It's a private company in Melbourne on stem cell immunotherapies for cancer. Delphi Krishna is Director of Cell and Gene Therapy Platform, R&D Strategy, Portfolio and Operations at GSK. Wave, wave, thank you. Keith Thompson's the CEO of Cell and Gene Therapy Catapult. Wave, Keith. Tian Tran, Director of Operations and CGMP Facilities at Stanford Laboratory for Cell and Gene Medicine. And Lisa Latanzi, Translational Program Manager for Therapeutic Genome Editing, Stanford University School of Medicine and Pediatrics. And Max Selman is a Client Relation at Aldevron. Right. So that's the panel. And so we're going to take a couple of minutes just to tell the panel, tell everyone here what their expertise is, so you've got some reference from that. So why don't we start from right this side, Keith?
So if you wanted to start with expertise,
yes.
It'll be short.
Yes.
It'll be short.
Be short, a couple of minutes.
For me anyway.
No more.
So Cell and Gene Therapy Catapult's based in London. It's been going now for 7 years. I set it up as part of a big initiative in the UK to help stimulate the growth and transfer of technology from universities into firms. And we've assembled a team of about 200 cell and gene therapy experts covering everything from regulation, health economics, but particularly process development. And we've built a very large-scale manufacturing centre in Stevenage, just north of London, which is a unique model. And I'll be happy to go into why that's unique and why it's been designed to help stimulate the growth of the industry rather than just serve kind of individual clients.
Thanks, Kate.
Right. So I'm with the Stanford Laboratory for Cell and Gene Medicine, and it's better known as LCGM. It's a purpose-built CGMP that is compliant to regulatory guidelines. It translates novel therapies and produces clinical patient products in early stage clinical trials. We have functional groups that have capabilities that kind of spans the whole arc of translational life cycle. So the capabilities includes GMP consultancy, GLP oversight, quality control, quality assurance, and a regulatory management of submissions. So we cover that whole range in an academic setting.
And Lisa?
Right, so I work at Stanford Medicine in the unit of definitive and curative medicine. We work on a variety of translational programs, and they are mainly constituted on gene therapy and genome editing protocols. I'm the project manager responsible for the genome editing area. I work directly in the Mark Porteous lab, which has developed the technology of CRISPR-Cas9 coupled with the AAV6 delivery for the genomic modification through the homologous recombination repair. And we have into our pipeline a variety of clinical trials that generate from scientific discovery and could possibly be translated for a therapeutic for patients. My role in particular is to interact with a variety of professionals and build teams that could synergize to perform preclinical and IND-enabling studies. Of course, I work in tight conjunction with the LCGM, the GMP facility. Together we perform technology transfer, process development, and we establish the final clinical protocol. And then I supervise as well timelines, budget, quality controls with the help of experts.
Okay. Thanks. Delphi?
Hi everyone, my name is Delphi Krishna and I'm the Director of Cell and Gene Therapy Platform at GlaxoSmithKline. I'm in the R&D Strategy Portfolio and Operations team, and my accountabilities are 3 to 5 year business strategy development and deployment, annual objectives, performance, operational model, strategic workforce plan, etc. GSK has a pipeline in oncology cell therapies, CAR and TCR products. As you may all know, we started out with rare diseases, which we divested to Orchard Therapeutics. And we have a group of around 300 people, about 250 in the platform, and around 50 in the oncology research unit.
Max?
Hi, my name is Max Selman. I'm part of the client-facing team at Aldevron. So a little about Aldevron. We're a CDMO based in Fargo, North Dakota. We've served clients globally from research through GMP, everywhere from really early stage companies up through people who have commercial products. And we manufacture biologics, so plasmid DNA, protein, mRNA, again, research through GMP. As well as antibody discovery. A lot of our clients are really heavily involved in the cell and gene therapy space, and our role is really to supply raw materials that they use in the creation of their therapeutics. So we heard Michelle in the last panel, she mentioned we've got some off-the-shelf plasmid products in addition to custom services. We've actually done the same with proteins. We have Cas9 Nuclease. GMP is a catalog product. We're the only company to do that. It's just a way to kind of support the cell therapy space, reduce barriers to entry a little bit. So that's just a little bit about us, what we're doing.
Thank you very much. Well, you've got all the panellists there now, so there are still seats down here if anyone wants to come forward. Please don't feel uncomfortable standing up. So I'm going to start with a question to the panel. How do you decide which outsourced services to prioritise or pursue first? What factors are driving your outsourcing decision? And let's start with Delphi because she's in the large pharma perspective.
Sure, thank you. So as far as the big outsourcing things are concerned in the CMC space, plasmid, cell banks, vector cell, at GSK we made the decision strategically to outsource plasmids and cell banks and vector and cell as well, GMP manufacture and testing, we do have outsourced relationships, but our long-term intention is to build capacity internally. And this was sort of intentional because as a drug development organization, we thought that our primary interest was in developing an organization of scientists and engineers who can translate therapies into the clinic, do the process development, the analytical method development, formulation, and make them IND ready. So that's where we think our expertise is, and we thought that we would build an organization around that. We have invested in our own clinical vector manufacturing facility, so our long-term intention is to exercise control in that space. but it's built mostly on our portfolio and the inflection points and when and how it makes sense under tight R&D budgets to do that.
Okay, and Lisa, what about the small program, academic perspective?
Yeah, so I can provide a perspective from a small program and also an academic environment. Of course, as I told you, we work in translational medicine, mainly in genome editing, so there are some key reagents that we cannot produce ourselves, and you want to start very early to introduce into your supply chain, like the GMP production for proteins such as Cas9 or synthetic guide RNAs. We also, as I told, use AAV6 vector as a donor template for the homologous recombination, and the AAV6, as we know, is considered a drug substance that has to be well qualified in the IND. So you might want to engage outsourced vendors pretty early with that once you have established your protocol, your manufacturing protocols. And the execution of the contract is strictly related to the funds and timelines. The budget availability for academia is something very struggling. We're always struggling with budget. And I guess it's quite requested to vendors and companies to provide flexibility for academia and clinical centers in terms of scale production and pricing as well. But it's crucial to insert and add to your manufacturing these reagents as soon as possible. I'm also referring to reagents such as consumable like medium and cytokines. It's better to introduce these reagents as soon as possible in your research too, so that at the time that you are providing your CMC, you already have all the reagents in place. That is something very important.
It must be nice to have a whole university there for researchers to be able to plug into that. Stanford, I wish I was there. We're a small startup company in Australia, and there's very little expertise outside, and this must be the case in some other countries as well, that this kind of provider expertise is not there. There's no viral GMP production facilities in Australia, nor is in any GMP materials available for CRISPR-Cas9, etc. So we've got to plan very carefully, and so once we know what our manufacturing systems are, then we have to actually find a partner in the US or wherever that will actually then work closely with us. And the problem of time differences, talking to people at different times, they're challenging, endlessly challenging, and not necessarily understanding import-export requirements that country-specific and so on. This can lead to very long periods of delay, and you essentially can't afford to have that, and it puts you at a big disadvantage. So the prioritisation is to sort of be extremely well organised and go out and create those partnerships with the providers as soon as you can. And then you get a personal relationship going with them so that they can understand what your particular problems are and what maybe your regulator as well is going to be concerned about, so that they can provide that. And we've kind of made many mistakes on this pathway, and it's actually cost us a lot of time to go back and repair it. And there's a lot of nodding going on here. That you can't afford to make these mistakes, because what it does is take that timeline out for you're actually getting anywhere close to patients a very long way. So then your investors are pressuring you because you're missing milestones, etc. So I think the prioritisation has got to be built around how well you've organised your development program and how well you've connected to the providers that you need, and that have that personal relationship. So you can ring them in the middle of the night if it's your middle of the day, and they won't be upset with you. And that means really getting that personal association. And I can't. It's just another dimension that makes it incredibly difficult unless you do that. We think that we're well organised, but clearly at times we miss a point, and that's going to be costly. So And if you make a change in the manufacturing, of course, then you really need to be in that connection to say, well, we need to make an adjustment. You know, we want to use AAV or something instead of lentivirus. And can you cope with that? Can you? So there's all of these things, and I suspect there may be people here who are experiencing those things. Again, any questions from the audience? I'm prepared to take at any time. So put your hand up, and we'll recognise you. Come down. Just here.
Do I need to turn it on? Is it working? Okay. I guess it's a question for Delphi. What are the considerations around outsourcing plasmids versus also bringing plasmid production in-house? You mentioned you'd bring in viral vectors and cell processing, but not plasmids. Why is that?
Yeah, that's a good question. I mean, for now, our plasmid production is, we have made the decision to keep it external. We are still thinking about long-term to bring it internal. And the main reason for that is the long lead times, the loss of control, and sometimes even quality issues with keeping things external. I think at the end of the day, it just boils down to there's a certain amount of money you're given to play with, and where do you put your bets? And we thought we would put it in the things which are harder to do, so to speak, scientifically, more complex, as opposed to plasmids, which relatively have been done for a long time. So that's kind of how we made the decision.
Okay, I'm going to move on to the next question, but again, anytime, please raise your hand. So what are some of the factors that influence your timelines, and how do you customise services and still meet client expectation for rapid turnaround? Let's start with Xian first, from a small academic perspective.
Sure. So being a small organization of only 35 and serving many investigators, we pride ourselves on our expertise in the realm of process development. But as you know, when the investigators come to us, they are at various stages of development of translational readiness, so to speak. And the last thing on their mind is actually spending time on process development. So, you know, priding ourselves on that expertise, we have very thoughtful conversation and dialogue with our investigator to provide them with the perspective of where they need to be as far as regulation, and just come to an agreement to what your target, simply your volume or the attributes that you want to get to. And so if you come to a kind of high-level expectation of each other, I think you can work out the timeline better. Connecting the dots between the different services and entity as far as the investigator, the CMO, and the outside vendor is very important. Having the interpersonal point of contact, you know, Annalisa and I work very carefully and thoughtfully together to then connect all the dots with our vendors and outsource. And that's how we manage timeline. You know, a lot of conversation.
That's what it takes, for sure. So Max, what about the perspective from a mid-scale CMO?
Sure, and you know, kind of jumping off of something Jen just made reference to, which is I think a big challenge is that on the custom side, development is often, you know, a moving target, and you know, a challenge for us as we get into these projects and start talking about what the process is going to look like, what the timeline is going to look like, what the cost is going to be. You know, a big consideration is how to kind of factor in that uncertainty of how development is going to proceed into a timeline. Speaking from my experience, at least, at Aldebaran, one of the services we provide is custom development for protein purification. So you have a custom protein you need to figure out how to make it. We can lay out a plan. We can lay out you know, in stages and say, hey, here's how we're going to go after it. We're going to try these chromatography conditions, then we'll move on to this. And at the end of this process, we'll have a purification method for you that's scalable. We can plan that out, but until we actually get hands-on with the protein, and really get into it, it's tough to say if that science is going to cooperate. And so really all we can do on the custom side is really be upfront about the challenges, be upfront about the risks, try to really rely on the fact that we have really strong, technically proficient scientists who can speak to data as it's coming out, and just really, keep updating timelines, keep updating prospectus, basically based on how the project is going. The one other thing I'd note, with regards to raw materials that go into cell therapy or gene therapy, there's certain things that are inherently customized. There are always going to be custom reagents, like a guide for a Cas9 that's target specific. So no matter what, you're going to go source that from someone, and that's going to be a long lead or a longer lead item. At Aldevron, we've tried to take in and make catalog products wherever possible, basically out of the components that are going to go into these custom medicines that aren't quite as custom. So Cas9 or a helper plasmid, things like that.
Great. Well, let's go to Keith, because you have a different system. You can
Sure.
have people enter your program and then do it with your assistance, but you must have a bit of variability there.
Yeah, absolutely. So the whole purpose of the Catapult is to stimulate the growth of an industry. So we approach everything from that perspective, whether it's assisting, the formation of a spin-out and taking an academic process and industrializing it. We have an industrialization group with 70 scientists in it who are working on generic platforms, and then using the skills they have on the platforms to actually apply them to whatever solution they've got. Or we're working with SMEs, or some very large pharma, in fact, to work up these technologies. So that may be anything from optimizing an individual unit operation, whether it's upstream or downstream, or kind of a molecular operation, if you like, or to a total one. And we would then transfer that back into the company, and that may in fact be an in-house operation, or we might help them take it to a CMO. But the really novel thing that we do, and what that is a kind of variation of what goes on in most places, except that we have the luxury of a very large grant to develop the kind of platforms. But the really unique thing we have is the large-scale manufacturing center in Stevenage, because when we were puzzling about how we were going to help accelerate the growth of firms, and this is in 2013, and frankly in 2013 there was hardly any contract manufacturing business, and there was hardly any firms. The sector was well served with academic production units, certainly in the UK and globally, but really there was no large-scale infrastructure out there that was, if you like, follow on space and expertise. And we talked to VCs about why they weren't investing in the space, and they said, we like the technology, but every academic group that we want to form a spin-out with, they want £25 million or $50 million or whatever it is to actually build a factory, and we're not in that business, they said. So we decided that after exploring lots of options, that we would build this center which essentially had ready-to-use infrastructure with very large clean rooms, not your average small academic clean room, but very large, 100 to 140 square meter clean rooms that could take scaled up processes, whether it was multiple autologous, and some of our collaborators are doing teens in one room, up to 1,000 liter reactor, viral vector, cell processing, anything. So we built this very, very large center, and it really suits firms that are operating at scale. And our objective is really to get them at scale so that they can get kind of pivotal trials done and initial in-market supply. And ultimately, they will learn not only the process that they use, but actually take the benefit of what we've done with putting this big GMP wrap around everything, whether it's the quality management systems at commercial scale, warehouse, supply chain logistics, and we have five well-known firms in the center at the moment. We have Autolus, Adaptimmune, Cellmedica, TCR Squared, and Freeline, covering cell processing, CAR-Ts, TCRs, Lenti, AAV, the lot, and we're just doubling that. And the firms are generally in that supported space for anything from 3 years to 7 years to God knows when. So these are very long-term commitments. They're not proceeding on a batch-by-batch basis. And the effect of that has been tremendous, because what it's done is it's helped unlock huge amounts of capital in the UK, but also really accelerate the growth of these firms so that they actually add value to their own to their own stock or what have you, by having really credible manufacturing operations. Gone really well. Stevenage is now the largest cluster of, it's right next to GSK, of course. Stevenage is the largest cluster of cell and gene firms in Europe, and within a few hundred yards, there's about, I don't know, 11, 12 firms and over 500 people at the last count. And you can't count them because they're just growing out the ground, you know. It's a fantastic and different operation.
So you've got four very different models here, haven't you? But I don't know what the final perspective is going to be for this space. Is it really going to be, you know, dropping into that kind of incredibly helpful entity, or really, are you going to go to the experts, either in academia or the CMOs or the large companies? It's a smorgasbord of choice, but I imagine eventually something's going to dominate the landscape, and I think it's probably a little early to decide what it is. Are there any questions coming from the audience? Anyone like to ask the panelists anything about what they've already said? No? Okay, but if you do, let me know. So let's move on to another set of questions. What are the risks you're taking right now with your current outsourcing setup? How do you identify areas of risk within your network of outsourced services? Maybe we can start with Annalisa and give us a perspective from what kind of risks are you taking there? None, or
Yeah.
the university allow you to take any risks?
Everything is at risk. No, seriously. So we are developing new technologies, and basically genome editing is something that's just starting to enter the clinics, not even the market, I would say. So our main struggle right now is to find good industrial partners that are capable of producing the reagents, because if you think how many industrial partners are capable of producing GMP, Cas9 or guide RNA, so it's really a sort of partnership with them. And these companies are requested to run process development to adjust the process for our product on purpose, customized. So there is a risk. The main risk is a production failure, delay, and quality failure. And the main risk mitigation is to engage as soon as possible with the expert leaders in the field that could really provide expertise for us, and communication and good management. I've seen projects that were in harder times with production, but the management was really good and could keep the timeline and resolve the problem. And in the end, we were successful.
Delphi, what about you? What's your perspective there?
Yeah, so in addition to timeline and quality, as you mentioned, I think one of the biggest risks that we are facing is turnover of talent.
Yeah.
Mm-hmm. So at our partners, oftentimes we are learning, they are learning. Talent is in short supply. So folks are learning a little bit, moving on to their next big opportunity. So it's a very, very challenging environment. And because talent is moving on so quickly, a lot of young people coming in from the academia into this environment, learning, moving on. the traditional things about quality risk management, the quality systems, data integrity. So a lot of risks around that area, just because people are moving around so fast and not absorbing, and the systems are not forming. So that's one of the big risks in addition to yours.
Max, your thoughts on this?
So, from our perspective of just kind of seeing what our challenges our clients are dealing with, one thing that I think comes up that, when we see this, it kind of is like a red flag that, something needs to be thought out a little bit better, but having just really a clear vision of what your end state should be, not having that would be a risk. So, I mean, in terms of scale, final quality specifications, things that absolutely absolutely have to be or absolutely cannot be in your final therapeutic form. These are all things that, if they're not well thought out as you get into the development process, certainly on the very front end, you're going to find that they percolate up just at really the wrong time, like right when you're getting ready to think about a tech transfer to GMP, and then you realize that you didn't have your process quite as fully understood as you thought you would. And so just right there, kind of having an understanding of the end goal. From the supplier side, I think something that's important, a risk that we take is kind of losing focus on what's really important here, which is patience at the end of the day. So like Annalisa mentions, going to a supplier and risking the timeline is delayed, and that kind of causes ripple effects. We can really risk losing track of what that actually means, which is that there's someone who's not getting a dose of potentially life-saving therapeutic medicine on time. So, it's something, for us, just really making sure that it's understood throughout the company, right? That like we're not just talking about making a product. We're talking about actually something that is gonna go in and help someone. Sorry, I bumped you there.
So Jian, are they all early stage companies or entities or members of faculty that are coming to you, or is there some difference in scale? Are there bigger scale entities coming in? Do you find any variance there in how you approach that? If it's a larger scale or a more developed entity, do you see that variance, or are you really always dealing with very early stage?
Yes, we do. But if I could add on to
Yeah,
that
sure.
last question of the two risks, two items that comes to my mind immediately and from experience is the field is trying to keep up. So the having a GMP facility, you understand what is required, and when you go to these facilities that are promising to produce GMP reagents, well, they're not set up for GMP manufacturing. And when you go to the site visit, definitely recommend that that's a requirement to go on site and see the facility that is being produced. There were a lot of risks when we saw this one particular service provider. But I guess the balance that you have to think about is then the cost. Are they a little less, but they're willing to partner with you through process development, but they're not quite set up. They're not set up with even their equipment or their facility. So the other area that I think is important to think about when you're weighing the risk is the knowledge of quality agreements.
Right.
That's so that's so important to keep in mind. It sets the communication. It sets the expectation between your service provider and yourself, so that you're not caught in when there is a product failure. How is that communicated? How are deviation handled, and how are they resolved? And how do you move on? Who is responsible for the cost of that failure? So I just added those two items there.
So what about the difference in development and scale?
Sure. Definitely there's a huge wide range between the scale, depending on if it's a rare disease, if it's phase 1 and 2. We were actually I guess, privileged to have gone involved in a project that we were at phase 2B and went to the FDA, and they told us, stop. You can write up a summary report for this, and it was after only 7 patients. and go to phase 3. We were ecstatic, but at the same time, like we're not set up for phase 3. We're a academic
problem to
GMP
have.
phase 1 and 2. But what that allowed us, in the leadership team, we talked about it, and I said, actually, I've been in Australia and we did phase 3. Why not? But we looked into it carefully and engaged the DMPQ and provided them with a plan and kept providing them with progress report of our our process of developing the processes and procedure to be able to handle a phase 3 for that particular project. So I think engaging the FDA as far as what is expected as far as scale is important, and what stages they're at. I mean, we're designed for phase 1 and 2, so I think that's our bread and butter. But to ask us to go beyond, that's a whole other scale.
So Keith, do you absorb all the risk for everybody in your organisation, or is it portioned in some way?
It's a really interesting question, because for this sector, the key thing that's come out from everybody is the sheer pace of development is like nothing I've seen before. I grew up in the monoclonal era, and we thought that was fast. It wasn't in retrospect. This is so fast, and the pace that people are moving at, it isn't really compatible with the kind of plodding way that biologics and drugs are kind of developed. Although for those that do the drug development in small molecules and large molecules, it probably doesn't feel like that. But compared to this, it's slow. So we try to partition the risk in the sector so that all governed with quality technical agreements. It's a risky business in a risk-averse world. So we have quality technical agreements, so everybody knows what they're doing. They're all well managed. And then what we do is we try to absorb and forecast where the next kind of levels of risk are, so that we're actually working on those independent of our collaborators. So I'll give you a few examples. The supply chains for CAR-Ts, TCRs, they are really poorly developed. And everyone thinks the supply chain around getting the patient material, that's only a tiny bit of it, okay? So in the unit that we've got in Stevenage, we have calculated that there will be 1.5 million materials movements that all have to happen in an orchestrated fashion, from the supplier of all of the plasticware, the reagents, and that all has to hit in 15 times every 2 weeks in a clean room for autologous or what have you. So we've actually put a lot of effort into getting off-site warehousing and kitting in a GMP environment so that this stuff can just flow through. And our collaborators, who are kind of pulling their hair out on their process, they don't have to think about, well, they do have to think about it, but we're doing a lot of the thinking for them and with them to make that easier. And then on the way out, of course, everyone's thinking, I've got to make this product, got to make this product, but the hospital end isn't set up. So we've put, along with the UK government, £30 million into developing advanced therapy treatment centres, where the receiving end of hospitals and the systems required to actually do this on scale, whether it's registries, IT, is kind of worked out. So it's all about partitioning the risk, and we'll take the risk on behalf of the industry that's appropriate for us to take, but we can't really take the risk on the individual products that the companies are developing.
Right. So, so there are a lot of, I think there are evolving other kind of programs. I've seen some things like the CCRM model is sort of, is a little bit like yours,
That's right.
but not the same. But there's an Innovate Bio program which is encouraging people to come in where they actually fund and try and help them do that. I mean, all of these things are in evolution. Do people feel that there is going to be a dominant type of provider eventually? Annalise, what do you think? Is there going to be a dominant type of provider, or are we going to sort of have everybody providing different things? I mean, will the university continue to provide that at Stanford because there's a big call for it internally, but you can make money by external inputs and things?
I mean, it depends on the level of which this therapeutic are developed. So if thinking about clinical trial phase 1, 2, you're still trying to prove safety and efficacy. So I think academics, clinical centers are on the front line for that. I know there are also some private sectors that are running their own clinical phase 1 trials. But thinking on a phase 3 and a commercialization, we need bigger providers, of course. And that will be interesting at the time of licensing some IP or having some partnership with the industry. That could be a very interesting scenario.
Right. Well, Delphi, everyone thinks of the bigger companies being able to sort of provide all of this eventually because they will take it into their business programs and sort out, you know, this and make, you know, have the providing happen. But I'm not sure whether the big companies like GSK and others are still think that kind of autologous treatments are a good business, or we really got to move to allogeneic type processes and so on. So
it depends on the disease at this point, I would say. There are some orphan drug disease that are really seeking for a therapy. Some others already have therapies, so the first step is to prove at least the same level of efficacy and a good safety to enter into the market. Otherwise, at the moment, for example, for new therapeutics, there is a comparison between gene therapy, which has already entered the market, some from disease, and now the upcoming genome editing. So we are in the interface. Genome editing has to prove to be more efficient, or at least at the same level with a similar safety. And I think the upcoming clinical trials will be crucial, at least to prove it for some.
I don't think the human immune system is going to kind of lie down and play ball just for the convenience of business models. So you know, the autologous and increasingly personalized medicine is still going to accelerate. So you would be foolish to just bet on one thing at the moment.
Yeah.
Well, we're nearly out of time. Unless there's some questions, I think please come and see the panelists and have a chat to them. I want to thank the panel for being terrific and thank you for responding in such a sort of positive and useful way. So thank you very, very much, panel, and thank you for being here, everybody, and participating.